Hi friends, welcome back to KPS Bio Tutorial. In the last class, we have been discussing about the organs of immune system. In organs of immune system, we were talking about the primary lymphoid organs, secondary lymphoid tissues or organs, tertiary lymphoid tissues, and we covered all the organs, tissues of the immune system in the previous episodes. The immune system consists of many organs, tissues, cells, as well as molecules. In today's episode, I would like to talk about the cells of immune system. I request all of you to subscribe this channel if you are not yet subscribed. Also enable the bell button so that you will get instant notifications when I upload the classes. I also request you to listen all the episodes in this series. For that I have given a playlist of all the classes here in the I button. So let us go to the topic, cells of immune system. Welcome back once again to KPS Bio Tutorials. Today's topic is cells of immune system. We know that in our blood there are numerous cells. They are called blood cells or blood corpuscles. The entire cells of the blood can be divided into three categories: white blood cells, red blood cells, and platelets. We know red blood cells or RBC, they are concerned with the transport of oxygen. It has nothing to do with the immune system. Whereas the platelets, they are concerned with the coagulation of blood. Whereas the white blood cells or white corpuscles, they are also called leukocytes. The red blood cells are also called erythrocytes. And these leukocytes or white blood cells, they are mainly concerned with the immunity. The white blood cells are again classified into two categories: the granulocytes and agranulocytes. Granulocytes include three cells: basophil, neutrophil, and eosinophil. These three cells belongs to the category granulocytes. As the name indicates, their cytoplasm contains numerous granules. Thereby they are called granulocytes. Whereas the monocytes and lymphocytes are said to be agranulocytes because their cytoplasm did not contain any granules. So these are the cells present in our blood. Among these blood cells, white blood cells belong to our immune system. This is the normal blood cell count in adult human beings. The RBC, red blood cells, composed of 5 into 10 raised to 6, about 5 million. Blood RBC are there in 1 millimeter cube of our blood. Whereas platelets form 2.5 into 10 raised to 5 per millimeter cube of blood. Leukocytes 7.3 into 10 raised to 3, about 7,300 in number per millimeter cube of our blood. Among these leukocytes, neutrophils form 50 to 70%, lymphocytes form 20 to 40%, monocytes form 1 to 6%, eosinophil form 1 to 3%, basophil form less than 1%. This is the proportion of different leukocytes in our blood. All these blood cells are generated from a common ancestral cell or a common multipotent hematopoietic stem cell, which is also called hemocytoblasts. They are found in our bone marrow. This multipotent hematopoietic stem cell has a potency to develop into different types of blood cells. Thereby they are called multipotent cells. You might have heard about totipotent cells, pluripotent cells, also multipotent cells. What do you mean by this potency? The totipotent cells are the cells which can form all the cell type in our body, including extra-embryonic or placental cells. The embryonic cells within the first couple of cell divisions after fertilization are the only cells that are totipotent. So these totipotent cells have a potency to develop into any type of cells in our body, including extra-embryonic cells or placental cells. Only few embryonic cells just after the fertilization. Only few embryonic cells formed by the division after fertilization are only totipotent cells. Whereas the pluripotent cells that can give rise to all of our cell types that make up our body, but it cannot form extra embryonic or placental cells. The embryonic stem cells are considered to be pluripotent cells. These pluripotent cells can develop into any cell type. It may develop into nerve cells, it may develop into muscle cells. In this way, it can develop into any category of cells in our body. Such cells are called pluripotent cells. Whereas the multipotent cells, it can develop into more than one cell type but are more limited than pluripotent cells. Multipotent cells have a limited potency to develop into a few category of cells, but it cannot develop into a broad category of cells. So this is about the potency. Our hematocytoblast or hematopoietic stem cells are belong to the category of multipotent cells. These hematocytoblasts may develop into two lineages of cells. It may develop into a myeloid stem cell or a lymphoid stem cell. This myeloid stem cell can develop into a megakaryoblast, which is then develop into megakaryocyte, and from which the platelets are formed. In this way, these cells can develop. Or the myeloid stem cell can develop into proerythroblast. From the proerythroblast cells, the reticulocytes will be formed. From the reticulocytes, the erythrocytes or RBC will be formed. Again, the myeloid stem cell may develop into myeloblasts, from which basophil, neutrophil, eosinophils will be produced. And from monoblasts, the myeloid stem cell may develop into a monoblast. From this monoblast cell, monocytes will be developed. From monocytes, macrophages will be developed. So these cells, the platelets, erythrocytes, basophil, neutrophil, eosinophil, monocytes and macrophages are the cells found in our blood. They are all developed from the myeloid stem cell. So these cells belong to the myeloid lineage. And this multipotent hematopoietic stem cell may also develop into another lineage, namely lymphoid lineage, or it may develop into a stem cell, namely lymphoid stem cell. From this lymphoid stem cell, the lymphoblast cells may develop. From lymphoblast cells, natural killer cells or NK cells, as well as small lymphocytes will be produced. And these small lymphocytes may again differentiated into T lymphocytes and B lymphocytes. From the B lymphocyte, the plasma cells develop. So these plasma cell, B lymphocytes, T lymphocyte, NK cells are cells present in our blood as well as in tissues. And these cells are called the lymphoid lineage cells, or they are derived from a common lymphoid stem cell. So this is the path of development of different blood cells. Among these cells, except erythrocytes and platelets, all others belong to our immune system. And now we can analyze in detail about the lymphoid cells. You know that they are developed from the multipotent hematopoietic stem cell in the bone marrow, from which the lymphoid stem cell will be developed. From the lymphoid stem cell, lymphoblast cells are differentiated. From lymphoblast cells, NK cells and lymphocytes will be developed. Lymphocytes again differentiate into T lymphocytes and B lymphocytes. B lymphocyte again differentiate into plasma cells. So these cells are called lymphoid cells. Let us Exam B lymphocytes. You are very familiar with the B and T lymphocytes. That is the two major types of lymphocytes. Now we can analyze B lymphocytes. The B of letter of the B lymphocyte stands for its site of maturation. In birds, they are matured from bursa of Fabricius. This bursa of Fabricius is denoted by the letter B. In the case of some mammals, they are matured from bone marrow. In that case also, this letter B stands for bone marrow. But it is first discovered from the bursa of Fabricius, thereby the letter B arises from bursa of Fabricius. About 1.5 into 10 raised to 5 molecules of antibodies are seen on the membrane of a single B cell. And all these antibodies have an identical binding site for an antigen. On the surface of the B cell, you can see numerous surface molecules. Among these surface molecules, the majority of them are antibodies. They are called membrane-bound antibodies. About 1.5 into 10 raised to 5 antibodies will be seen on the surface of B cells. What is the role of these antibodies? These antibodies or membrane-bound antibodies will interact with antigen, as well as they can interact with the T cells and macrophages. This interaction induces the activation and differentiation of these B cells and for further development of the immune response occur. So the most important role of antibodies on the surface of the B cell is to interact with antigen in different ways. It may be either directly or through by interacting with the macrophage or with the T cells, and thereby these B cells get activated and it will go for the further development of immune response. And apart from the antibodies, numerous other molecules also express on the membrane of the mature B cells. We call a B cell matured when only they can interact with an antigen, or all the surface molecules are perfectly expressed on their membrane, then only the B cell is said to be a mature B cell. On the surface of these mature B cells, different types of molecules are seen apart from the antibodies. Among these, one is B220, that is a form of CD45 molecule. It is frequently used as a marker for B cell and their precursors. However, unlike antibody, it is not expressed uniquely by B lineage cells. It is a marker molecule on the membrane of the on the membrane of the B cells. It is also called CD45, and it is used as a marker, that means to identify a B cell. That means if all the B cells express this B220 molecule, so we can identify this B cell from the presence of this marker molecule B220. It is also called CD45. Then another molecule present on the membrane of the B cell is class 2 MHC molecule. It permits the B cell to function as an antigen presenting cell or APC. Class 2 MHC molecules are a kind of MHC molecule. MHC means major histocompatibility molecule. We will study the structure and functions of MHC in detail in coming chapters. So this class 2 MHC molecule is exclusively found on antigen presenting cells. So B cell is an antigen presenting cell. So using this class 2 MHC molecule, it will present the antigen. The B cell can endocytose the antigen, and the endocytosed antigens are processed and then presented with the help of this class 2 MHC molecule, and this presented molecule can interact with the T cell, and in this way that through this class 2 MHC molecule a B cell interacts with T cell, and thereby T cell gets activated by this interaction. Then CR1 or CD35 and CR2 or CD21. These are receptors present on membrane of the B cell for certain complement products. In our immune system, there is another system, namely complement system. This complement system consists of numerous proteins. So this CR1 and CR2 are the receptors to interact with the complement proteins of complement system. That also we will study in coming episodes. So here you can see numerous molecules on the surface of the B cells. They often referred as CD45, CD21, CD35, etc. Then what is this CD? CD means cluster of differentiation. It is also called cluster of designation or classification determinant. This CD system is a protocol or a system used for the identification and investigation of cell surface molecules. It is a system of identification, classification of and investigation of cell surface molecule. These CD molecules can act in numerous ways. Often they act as receptors or ligands. Some of the CD molecules act as receptors, some others act as ligands. Then what is these receptors and ligands? You might have heard about receptors, many hormone receptors and many receptors in the cell signaling pathway, etc. So ligand means is a substance or a molecule which will bind on the receptor. Usually the ligands are smaller than the receptor. So that is, if A is binding on B, then A is said to be ligand and B is said to be receptor. This CD system is commonly used as cell markers in immunophenotyping, allowing cells to be defined based on what molecules are present on their surface. So using this CD system or CD protocol, the cell surface molecules are studied, and based on that study, the immune cells are characterized and they are named and identified, and all these things are together called immunophenotyping. So the cell surface molecules present on immune cells or in any other type of cells are utilized for the characterization of the cells. Here you can see different types of cells and the different CD markers or molecules present at the cell surface of these cells. The stem cells consist of CD34 plus CD31 minus CD117. Here you can see in some region it is said to be plus and otherwise it is denoted as minus. Plus means this particular cell possesses this marker, this molecule, that is indicated by the plus sign. Minus sign is denoted as this particular molecule is absent on the membrane of this particular cell. And here you can see different types of cells and different CD markers. Here the T lymphocyte possesses CD45 plus and CD3 plus. That means T lymphocytes are said to be CD45 plus, CD3 plus. That means both CD45 and CD3 molecules are present on the membrane of T lymphocytes. So looking at the surface molecules, we can identify the particular cell types. The other molecules present on the membrane of the B cell is FcR2 or CD32. It is a receptor for IgG, that's a type of antibody. IgG type antibody receptor, that is called FcR2 or CD32. It is another molecule present on the membrane of the B cell. Then B71 or CD80 and B72 or CD86. These are molecules that interact with CD28 and CTLA4, that is important regulatory molecule on the surface of different types of T cells. On the surface of T cells, there are certain membrane molecules, CD28, similarly CTLA4. They interact with the B71 and B72 or CD80 and CD86 of the B cell. So B cell interact with the T cell with the help of these B71 and B72 surface molecules. Then CD40 is a membrane-bound molecule on B cell which interact with the CD40 ligand on the surface of T helper cell. Here you can see CD40 on the B cell interact with CD40 ligand on the T cell. So through the CD40 it interact with the T helper cell. So this is about the B cells. They are said to be B cells because they possess these type of surface receptors or surface molecules on their membrane. Identifying these markers or molecules, the B cells are get distinguished from other cells. So let us wind up today's class here. I think all of you understood well about this topic. If you understood the topic, you like the video and you make your comments in the comment box. I will be coming back with remaining part of this topic in coming episodes. Till then, goodbye to all of you.